by the different fish. But we also have some really wonderful genetic tools that have allowed us to ask questions about, well, what is the basis of these morphological changes? And one of the case studies is now understanding how marine stickleback, which have a lot of lateral plates, when they moved to freshwater, lost those plates. So these are it's a really exciting system to be able to start to ask, well, what is changing and what is the genetic basis of those changes? And something that's been kind of missing from this picture, people have been studying it, but I think there's a lot more to look at, is that, well, you are what you eat. And so as you start eating different prey types, you start getting exposed to a lot of different parasite communities. And so whether you're eating things in the water column, you're going to be exposed to things like the cestode, or if you're eating things off of the benthos, you're going to be exposed to a lot more insect-related prey, or things like these nematodes. And so across the past decade, Dan Bolnick has been sampling lots and lots of populations on Vancouver Island. And he's been finding some really remarkable trends. So in the case of the cestode, which has been the focus of my studies, across years you find really repeatable patterns of prevalence in these populations. And it's really highly variable. Some populations, you never find the cestode, and other populations, it's in almost 80% of the fish. So the question is, what drives this pattern? Presumably it's probably got something to do with environment, where there might be fewer cestodes present in some of these lakes, but for many reasons we think that that's not entirely likely, because it's a pretty small island. There's a lot of exchange internal hosts of parasites. Another possibility is that there's actually a heritable component that generates this pattern. And so that's what we went ahead and tried to test by sampling fish from both a high infection lake and also fish from a no infection lake. And the idea here was that if we bred a bunch of fish, took their offspring, brought them into the lab, and raised them in a common garden, we'd be able to ask, well, do these trends hold up if we infect fish in the lab? Do we see that no-infection lake fish do really still, are they resistant to infection? We were also able to generate F1 hybrids between these two populations to ask if there were any genetic patterns popping out, such as additivity or dominance. It's also important to know a little bit about the life cycle of the parasite. So like many parasites, it's a complex life cycle that involves multiple hosts. But the most important part of this is that this Schistocephalus solidus, the tapeworm, it has to get into threespine stickleback. It's an obligate intermediate host. So this sets the stage for some really interesting coevolutionary dynamics between the two, where stickleback are always encountering this tapeworm, and a tapeworm has to go through a stickleback. The other thing that really excited me about this system was that we're able to do this all in the lab. So we can take tapeworms, put them into nylon bags, and breed them in the lab. We can then infect them into copepods in a very rigorous way, and expose tapeworms to set amounts to expose fish to set amounts of tapeworms, and then assay a bunch of infection variables in those fish that have been infected. So this has been an enormous project. It's gone over 3 years, and it's involved a lot of people. I want to especially point out that my collaborator is Will Shin, who's now a graduate student with Dan Bolnick. He's actually up in Vancouver sampling fish right now. And Dr. Natalie Seidel, who's in the audience and who's giving a talk on Tuesday, I encourage you all to go to if you want to learn a little bit about how tapeworms might suppress the immunity of fish. And I can't talk about all of this today. It'll be out soon, but what I will tell you about is some data on the worms that are coming out of these fish, the mass of the worms, the total number of worms per fish, that is infection rate, so what the worms look like, and also something about the immune responses in those fish. And importantly for the methods, we exposed each of these fish in the following slides to 5 worms, and then we sacrificed the fish at 42 days, 42 or 43 days, and so everything is standardized along those lines. So the first question, do no-infection fish actually resist tapeworm infection in the lab? No, we found no evidence of that. So whether you're looking at high infection or no infection fish, we found that even in the no infection population, although we didn't see a whole lot of infections, there was no difference, about 13% in the no, 18% in the high population. No significant difference between the two. So, okay, it wasn't that simple, but when we started looking at the worms, something else popped out. So there's an enormous difference in how big the worms grow within the two hosts. Worm size is an extended phenotype of stickleback hosts. So from the no-infection population, the worms are almost microscopic. It's really hard to find these things. While from the high-infection population, they grow to be enormous. And so this is quantified. You can see the quantification of it here. And then when we actually look at the hybrids, we find that they, although they're intermediate, they're largely the same as the no-infection worms. There's something that apparently somewhat of a dominant genetic effect going on here. And I say genetic again because these are fish that have been raised in a common garden environment, and yet we're seeing reliable differences that are different among the populations. So, what inhibits worm growth? This gets a little tricky because unlike model organisms like mouse or even in humans, we know a lot about the immunology of these guys. We don't know so much about fish immunology. It's a growing field, but this doesn't mean that we can't make some educated guesses about what's going on. And one of the responses, immune responses, that's conserved across a lot of vertebrates appears to be this respiratory or oxidative burst response, which is when you produce toxic reactive oxygen species, ROS. These are radicals, free radicals, that are basically spewed over the parasites and damage them. And so within the fish, there's a lot of specialized, or within any vertebrate, there's specialized cells that produce ROS. I'm going to talk specifically about granulocytes. And the idea here is that our hypothesis going in was that, well, maybe if ROS is associated with any of these differences in natural infection prevalence, then maybe we have high ROS expression in the no-infection population because they're using it to combat the worms. Or maybe the high infection population has decreased ROS. It's entirely possible that there are high costs of mounting immune responses, and you might actually turn it off in some populations too. The way that we did these assays was that we took the fish, we took out their head kidney, which is a small organ, it's the main immune organ in threespine stickleback, ground it up, put it into cell culture, and then we exposed those cells to another chemical called DHR. The neat thing here is that when you expose ROS to DHR, you get a fluorescent molecule. So we could then take those cells, push them through a flow cytometer, and ask, well, what proportion of cells had ROS were producing it, and how much ROS was being produced by those cells. So we went ahead and did this, and we found some interesting results. So in the first case, we found that the no-infection, pure no-infection fish had lower total percentage of cells that just had standing levels of ROS going in. It wasn't a dramatic difference between the two populations, but it was significant, and the two hybrid populations were intermediate between the two pure strains, suggesting a genetic influence there. But really more striking is that they produce 4 times more ROS in their cells from the no-infection population than from the high-infection population. So this is really a plausible way that these fish might be combating or resisting the tapeworms, and perhaps explaining some of the natural variation that we see. I also want to point out that in these patterns, there was no difference between no strong difference between no-infection and high-infection fish, suggesting that these are kind of things that are just standing and aren't a byproduct of infection. So what I've just shown you are a bunch of correlations. The idea is that, well, we find some ROS variation, and that might be associated with natural variation. And we find some variation in worm size, and that might be associated with natural prevalence. And maybe even ROS affects worm size. But how do we go past correlations to causation and actually say that these are the things that are driving natural variation? We've generated a ton of F2s. We have about 700 animals that we've now infected or exposed, have 250 infected fish, and I just last week got the DDRAD gene sequences back for all of the animals. So I can't show you QTL, I hope I will be able to show you it soon, but we have some really strong patterns that have already come out. So if you look just at either intercross fish, which are F1 by F1, or back crosses to the no-infection population, we find, interestingly, a really strong association, negative association, with more ROS actually decreases infection prevalence. Although we didn't find any strong differences in the parental population, there was a tendency there. It's entirely possible that ROS might actually allow these worms to resist infection. Also, interestingly, we found no connection between worm mass and ROS, suggesting that these two are independent traits that are evolving within each of the populations. I'm also really excited about some of the physiological findings, what's happening during infection. So we find in a lot of these fish, well, specifically only in the back crosses to the no infection and the inner crosses, pus sacs. Sometimes those pus sacs are moving with live worms, sometimes they're filled with dead worms. It's clear that these fish are able to kill the worms using these granulomas. But there also appears to be costs. We find that in a lot of cases, the fish that are having pus sacs also have a fibrotic phenotype where their organs are entirely fused. And we've now just also been able to show that, at least in the case of females, females that have fused organs don't become gravid. So there might be interesting costs associated with differences between the sexes here. And now that we've gone out into nature and started looking at this more and more, now that we have a phenotype that we're actually able to know is associated with infection, we're finding this in nature also. And so I think that these things might very well allow us to go back and understand how this picture is coming together. And just a little bit of ongoing work. I also think this is really exciting. I told you at the start that we know the direction of evolution here, where marine fish are colonizing freshwater, and that so all the freshwater populations are their ancestors. Well, we've now started assaying marine fish, and it turns out, well, in nature they don't get infected with the tapeworm because the tapeworm can't live in marine environments. But when we bring them into the lab, we find the marine fish are entirely susceptible. Every time that we give them worms, they get hammered. But in every freshwater population that we look at, even in the ones from the high infection, there's a dramatic decrease here. This includes fish both from Canada and from Europe. It's a global trend that we're finding, that as you're moving into freshwater, it appears that you're evolving resistance to this tapeworm. And I'm also excited by the fact that when we look across populations at just infection rate, you find that low infection populations have very low worm mass, and high infection populations have very big worms. So I think that the trends that we're finding in the lab are giving us some kind of picture to go back out in nature and understand what's going on. And once we start getting some of these genetic loci, if you have a chance to run to Dan's talk in a few minutes, he's going to talk about all of the other expression data and other immune data that we're collecting. I think we're really starting to get a handle on the adaptation between the stickleback and we're generating resistance to this particular parasite. So with that, I just want to throw out some acknowledgments. Of course, Dan for being a wonderful mentor and giving us the opportunity to do this, Natalie and Will, and everyone else that helped us breed the fish, our funding sources in Canada for letting us take their fish. With that, I'll take any questions.